people talking.
Yeah,
Amazing.
I mean, we regard cancer as a major challenge, and of course it is. There's 1 in 3 of us will get cancer in our lifetime, but cancer is mostly a disease of the elderly. What we now need to do is to try and find out why some people are more prone to cancer than others and deal with it early before it becomes a problem. But it's said that you have the cancer for at least 10 years before it's clinically obvious that you've got it. So cancer obviously is something that starts much earlier in life than when the cancers become obvious.
Is it always there as a dormant from when you're born, or is it?
No, I don't think so. It's just that some cells in your body go wild, if you like, and they can sit there for 10, 20 years not doing very much, and then suddenly decide that they're going to grow and expand and become an obvious clinical cancer. But the changes probably occur in your 20s and 30s. You know, we're always being exposed to things that damage our genetic information. Stuff in our food, stuff in the air, stuff, radiation in the lab even. And anything that gives the cell a chance of making a mistake when it next divides itself could be the thing that eventually leads to a cancer.
Because that's the process, isn't it? Something goes wrong.
Yeah, something goes wrong with a cell. Its genetic program goes wrong. And mostly what happens is the program says divide when the cell shouldn't divide, but it also usually acquires new properties in doing that. So the two things that define cancer are cells that divide when they shouldn't and cells that travel where they shouldn't. And if you've got a population of cells that will continue to divide when they shouldn't do and also can travel through your body, that's when cancer becomes obvious.
Do they just pick a ride in the bloodstream?
Every cancer is different. Some of them will just spread, put their tentacles out, if you like, and move locally. Some of them will go in the bloodstream and travel to elsewhere in the body, and then that's where they sort of put the roots down and cause problems. So it's not one set of problems that makes the cell a cancer cell. It's several, but the two things that really make a cancer cell into a cancer cell is when they've got genetic damage, so they continue to divide and grow when they shouldn't, and they go places they shouldn't.
Just there's a little bit of noise in the background.
You're going to cut those too?
Excuse me, please. Hold the roll.
Ian Frazer too.
Pretty exciting.
Okay,
Okay, I'm going to leave off the cancer. I'll come back to cancer, but
yeah.
you and science, why? How?
Look, my father was a scientist, and he was a biochemist and ran a diagnostic laboratory. He introduced me to science, I suspect, when I was about 10 to 12 years old, by buying a chemistry set. In those days you were allowed to have one of those at home, and you could do all sorts of things with it. But I also was inspired by one of my teachers at school. He was a physics teacher, and together with the magazine New Scientist, which still exists, but it was brought into existence about that time, got me excited in what science was all about. And I was fairly sure I was going to be a scientist, but the interesting thing was I did all the right stuff at school so I could go to uni. Went to university to study astrophysics, which I don't think my father thought was a particularly good idea. And I worked out pretty quickly it wasn't either, because there was very few jobs for astrophysicists, and they almost all were in CERN in Switzerland, and that wasn't really what I had in mind. So that I changed to study medicine at university, with not with research particularly in mind at that point. I just wanted to become a doctor. But halfway through the course, I met a couple of people who were inspiring. A guy called John Habishaw, who was a scientist who was very interested in a cell called macrophages, called a macrophage. I think it's a big cell that eats things. Literally translated, and that was just at that time, that was a very hot topic. This idea that you could, well, as George Bernard Shaw put it, stimulate the phagocytes. In other words, make the macrophages eat the cancer cells. And so I did a year's honours project trying to make macrophages eat cancer cells. I didn't succeed, but it got me interested in the science. And when I finished my medical training, I decided that I would become a scientist at that point, but I was going to be a doctor as well. I came out to Australia for the first time when I was halfway through my medical course. We actually had, the university required us to take a trip somewhere outside of the United Kingdom. And I came to Australia to work with a guy called John Habishaw down in Melbourne. And he was interested particularly in how genetic damage occurs in cells. And so I spent some time with him and some time with Ian Mackay, who was the professor of medicine at that time. And between the two of them, they got me really hooked on the idea of medicine and research. But I had to finish my medical course in Scotland, and then I worked as a renal physician for a couple of years. And then skiing gets tied into everything, but it gets tied into this story too. We went off on a skiing holiday, and when I came back, there was a telegram under the door. Now, nobody gets telegrams these days, but the telegram was from Ian Mackay saying, "We were expecting you, why aren't you here?" And so I had to make a decision at that point whether I was going to go and take a science job at that point or stay training as a renal physician, which was what I was doing in Scotland. And Australia won. So I and my then pregnant wife came out to Australia for a 2-year visit, and we're still here. So that was to the Walter and Liza Hall Institute in Melbourne, which at that time was the leading immunology facility, I guess, worldwide in terms of research. And I got interested in whether you could take the immune system and use it to fight cancer. It was back to the same ideas I had with the macrophages and eating cancer cells. But this was a little more sophisticated because we had learned a lot more about how the immune system can be targeted to particular problems. And so that started me, if you like, on a career where I wanted to understand whether we could use the immune system to protect against cancer.
Could you paint me a picture of what it was like? And I'm assuming this is the 80s.
Yeah.
That you were doing this. Like, where were we? Like, on the graph of where we are today compared to where we were there, I kind of know the answer. What's happened in the last 30 years has been nothing short of spectacular.
Yeah, in those days there were an awful lot of things that we can do now that we couldn't do then. We were not good at genetics. You couldn't look at the genetic information in cells easily. We couldn't grow the cells that easily either, and particularly we couldn't change them in the laboratory to do different things. We didn't have monoclonal antibodies, so we couldn't tag cells to find out what they were. The technology of making antibodies that were specific for one thing was very important because it allowed you to characterize the cells you were working with. And much of even the technologies that we had available at that time, I worked with one of the very first flow cytometry machines in Australia as part of my work, and that was one machine which now every lab would have 10 of them, you know. So the technology has driven the science. And I think that's something that's important to remember, is that technology drives science. You can't do it. I mean, you can have the ideas, but if you don't have the technology that allows you to answer the questions, you won't get the answers. So what has changed over time is this tremendous ability to access better information about what you're working on. If you've got an individual cell, you can sequence the genetic information inside that cell. You can sequence the RNA, the blueprint for the cell to behave. You can see whether it's changed or not, and then you can change it and make it so it's not the same as it was before and see what the effect of that might be. And then we can grow these cells in culture now. We can do things with them which we simply couldn't dream of doing when I was a student or when I was a young scientist. And it continues to get better all the time.
Well, what are the big ones? From the 80s to today, so that's what, 40 years?
Now,
Do they ting off in your brain as they came through as you were learning and expanding? Do
Well-
certain things, certain breakthroughs still resonate with you? Go, wow, that was that.
Yeah. Oh, the very first thing that really changed what we could do were monoclonal antibodies, because we could, for the first time, we could take a population of mixed cells and sort out what was actually going on inside them. The biochemists at the same time were beginning to learn to look at the DNA inside the cells, which was the blueprint for that cell, but it enabled people to find out if there was a change in the genetic information in the cell. We could sequence DNA for the first time and in large quantities. All of these were basically machines that became available. You know, the technology has always driven things. The microscopy that we have now, we can look at living cells under a microscope for long periods of time and see what's going on with those cells. Whereas in the old days you could only take the cells, kill them, stain them and have a look at them. Now we can actually watch them doing what they do. And you can do that in a living animal now. You know, you can have a living animal anesthetized and watch what's going on with the cells in that animal. All these things make it possible to answer the sorts of questions that people ask about cancer or indeed about any disease. But I suspect that most people would say the thing that's been the major change is the genetic revolution. Not only can we sequence genes, we can make genes and we can put them into cells and see what they do, so that we can try to emulate the problems that arise in a cancer cell and see if we can understand what's going on. And it's not just the genetic information that's in the cell, it's actually the flow of information from the cell's nucleus, from the DNA to the cytoplasm of the cell, and the programming of that cell to behave or misbehave. All these things would have seemed impossible in 1980 and are now routine in 2025.
So the old days is 40 years.
Yeah.
It's not that old, is it really?
No, within the lifetime of most scientists who are alive today.
So really cancer research before World War II was minimal?
Yeah, the cancer research, we didn't really understand cancer as a disease. It goes back a while. Mechnikov understood that cancer was a group of cells that weren't behaving themselves, and that's more than 100 years ago. But he didn't know what was wrong with them because he didn't understand the normal cell biology. You know, the two things have to move in pace. If you don't understand how normal cells behave, you don't really understand how cancer cells are different. But all of that that's all post Second World War. It's mostly post 1970. I mean, the big breakthroughs came in the biochemistry, in the monoclonal antibodies, in the sequencing of genetic information, and it was spread by stealth, if you like. I mean, these things they don't happen in big chunks. They just gradually you accumulate more knowledge about how things work, and people work on different aspects of things, and the bits start to fit together. We still don't know entirely what cancer is all about, but we certainly know what a cancer cell does and why it does it and how it's programmed now. And that means that you can start talking about specific therapy for a cancer. In other words, that person's cancer is wrong because of this particular set of changes, and we know how to deal with those. It's not yet at the stage where we can just plug in the computer, so to speak, and ask it how best to deal with that particular cancer cell, but it's getting closer to that now.
Do you think that will come?
The answer is yes, in a way. I mean, we all carry abnormal cells around inside ourselves. I mean, from the day you're born, your cells divide, and when the cells divide, there are potentials for mistakes. Mostly the mistakes kill the cell, that's fine. Some of the mistakes will make that cell have increased potential to become a cancer down the track. You and I probably carry lots of clones of cells inside ourselves which are on their way to being cancer, but we won't live long enough for them to actually get there. You know, we have a use by date, and we can control cancer to the extent that the cancers never become real by our use-by date. I mean, the absolute use-by date seems to be about 120 years for the biology at the moment, and I think there's one person who's reached that, one woman. And for men, the cut-off date seems to be when you get to about 110. And if we can just stop cancer becoming obvious before you reach the 110, if you like, we've done a good job, because that person may carry cancer cells, but they're not going to get sick or die from cancer.
Somebody suggested to me that the reason cancer's become such a focus and something to be cured is because medical research has been so phenomenal in killing other diseases that knocked us off first. Would you like to comment on that?
Yeah, essentially that's true. I mean, leaving aside a few more recent viral infections, we're actually pretty good at controlling infectious disease. But the biggest killer is actually cardiovascular disease. And that's largely because we don't look after our diet and our weight, but it's also genetically determined to some extent. If you go to countries where the median age of death is in the 50s and 60s, then cancer is not a problem. Cardiovascular disease is becoming a problem. As you live longer, so the chances of you getting cardiovascular disease increase, so also your chances of getting cancer. I mean, there will always be cancers that spring up unexpectedly in young kids. These are cells where the cells have been misprogrammed, if you like, and instead of trying to make brain cells, they make a brain tumor, so that will always be part of the scene, but that's rare. I mean, these sorts of cancers are rare. The common cancers, skin cancer, for example, which if you live in a sunny climate, everybody gets skin cancer if they don't have a pigmented skin to start with. If you've got a pigmented skin, then you've got a big advantage. You won't get skin cancer. So these things, it's driven by the population genetics. You know, if you like, evolution has tried to produce the best possible means of producing a human. You know, and best possible as in will live a long enough life to make sure they rear their children, that's really what drives the whole system. It's successful reproduction. I mean, that's true for all animals, basically, and humans are not exempt from that requirement, so that we reproduce in our 20s, 30s, 40s, and it's okay then, from the point of view of evolution, and to have cancers occur when you're 60, 70, 80, because you'll have brought up and reared your kids and the next generations there. So that there's no strong genetic selection against getting cancer in your later years, but there's a pretty strong genetic selection against you getting it young. Is that?
I don't know, it's great. You've just made me think. I don't like that.
Yeah, well.
You speak about smallpox and things that came before, but I want to go back to, okay, you went from astrophysics. Now, I can only imagine that's in the tail end of the space program that may have inspired you.
Yes, to some extent. Obviously, they were flying people to the moon, but it was also this whole question of what's out there. You know, you look at. I mean, anybody who isn't inspired by looking at the heavens on a dark night, clear skin, you know, it's you expect to be asking, well, what's really out there? Are we the only people around? I mean, how on earth has this particular planet got billions of people living on it, and we don't hear from anybody out there at all? They're maybe all too far away, but that's just a technology issue. You know, you should in principle be able to overcome that and find out where other people are, or other somethings. I mean, we use the word people very loosely, but I think that I was interested in the concept of what was going on. You know, how do stars work? How do they form? How are they so different one from another? You know, they got all the physics there is really strange and puzzling, and we still don't have answers to it. We can't answer these questions without being out there, and we don't have the technology to do it. But as interesting as it wasn't people interesting, if you know what I mean. I was much more interested in people. I guess that's why I switched from astrophysics to medicine.
But you were studying to be a clinician.
Yeah.
And then what was the siren call of research then? Was there a trigger or?
Yes. Look, when I was in the medical renal unit in Edinburgh, there was an epidemic of hepatitis B virus infection, and that struck me as, people died from this, and I thought, well, you know, we should be able to prevent this. I mean, we had vaccines against polio and other things. Why not a vaccine against hepatitis B? So I talked with some people from Merck who were working on that, and I realized there were quite a lot of other vaccines that we really ought to be thinking about as well, to stop not just infectious diseases but cancer. Obviously my own interest was in papillomavirus, and that was inspired by the fact that the papillomavirus at that time was just beginning to be realized as a cause of cancer. The clinic I worked in Melbourne, I was interested in hepatitis B, so I got all the men who had sex with men, because they were the major carriers of hepatitis B, and I was trying to work out whether they were protected against a second infection with hepatitis B virus, which they seemed to be, but not always. And it was partly because we didn't know the best way to describe that hepatitis B and say, well, you know, we didn't have the test. There were other hepatitis viruses, and we weren't sure which ones were which. But there, while working in that clinic, the first AIDS patients started coming through from the young men who had come back from the United States, and they had acquired immune deficiency syndrome. They also had acquired the ability to get trouble with papillomavirus infection, with warts. And I couldn't work on the HIV because there were restrictions on working on that at the time, but I could work on the warts.
What was the restrictions?
Oh, just that you had to work in an environment where you weren't going to give other people HIV, and the labs that I were in weren't set up that way. So I couldn't do that work, but I could work on papillomavirus, and I thought that.
Explain from the get-go as a virus. What's its?
Papillomavirus is a virus which, it's actually a whole family of viruses. There's over 100 different types, but one broad group of them cause warts on your skin, and they're pretty obvious and pretty harmless. And then there's another group which seem to be associated with cancer, and particularly genital cancers, and particularly cervical cancer. So when I moved from Melbourne to Brisbane, I took my interest in papillomavirus with me. That was the virus I was going to work on, because I wanted to work on one specific problem. And the field for hepatitis B was well catered for at that time. There were plenty of people working in that area, and there was indeed a vaccine under test. But I got interested in papillomavirus and its ability to cause cancer. First of all, in the men who had sex with men, because they had a problem with their immune system because of HIV, and they got a lot of problem with the viruses which we now recognize as causing cervical cancer. So that told me straight away that the immune system could handle these viruses for most people, because if they didn't have an immune system because they had HIV, then they got problems with HPV, and if they didn't have HIV, they didn't seem to have much problem with HPV.
What's HPV?
Human papillomavirus. So that was why I took that interest with me when I came up to Brisbane, and I decided I would work on that virus, and was fortunate enough to meet up with a guy called Chan Su, who was also interested in working on that virus, and he came and worked in my lab. He and I met in Cambridge in the UK, where he was working on a different virus and I was working on a different virus, but we both combined our interest in papillomavirus and decided we were going to see if we could work out at least the immunology behind that virus, which we eventually did to some extent, but along the way we came up with a way of making the material which became the vaccine to help prevent papillomavirus infection, which was a bit of a surprise. We didn't set out to do that, but we got there anyway. And that's quite common in science. You start trying to solve one problem and you end up solving another one, which perhaps would be more useful.
So I heard you say that you were drawn to each other because you were both foreigners and hang on. Sorry about that.
That's all right.
I think you said lightheartedly attracted to each other, your partner, not your wife, but your laboratory partner in Cambridge, because you were both foreigners. You were Scots and he was Asian.
Yeah, that's true. I mean, well, we were different, you know, and I think because we had travelled to get involved in that, you know, our enthusiasm level was high. You know, we put a lot of, if you like, a lot of effort into getting to where we got to. And so we thought might as well put the effort into the science as well. And it was a puzzle. You know, there was obviously a story there. What we didn't immediately know was how many different sorts of papillomavirus there were. We thought there were two. Turned out there were quite a lot more than that. But there were two different groups of them, and we worked on the one which looked like it was causing cancer. And my aim was always to understand how the immune system would fight the virus. Jan's was how the virus caused cancer. And that worked very well together because what he did helped me and what I did helped him. And the net result was that we came up with a technology which became the vaccine against cervical cancer. We didn't really think at that time it would be quite as stunningly successful as it's turned out to be, but we could see that the body's defences could fight this virus, and we just wanted to see how they fought it. And we were thinking, well, maybe it will be these cells which were new at that time called T cells, which kill cells that are infected with virus. But it turned out that it was much simpler than that, and the best way to stop the virus, the papillomavirus, was with antibody, which is a very well recognised means of making vaccines. So the only real surprise in that was the technology to make the virus-like particles, which became the basis of the vaccine. We knew if we could get the virus-like particles, we would probably get a vaccine, but we didn't know how to make the virus-like particles, and that took a bit of research to work it out.
Okay, would you indulge me? What can you tell me of the history of vaccines from the get-go to where we are today?
Oh, look, I mean, if you want to go back in vaccine history, the obvious place to start is with vaccinia, with smallpox, because two important experiments of which you wouldn't get allowed to do today. One was you could use cowpox to protect people against smallpox. The two things looked the same, and one was in a cow and the other was in a human. Well, let's try it in the human and see what happens. And so a cowpox vaccine worked. And the other way that they did it was to give you small doses of smallpox virus in the hope that instead of getting a really bad dose of smallpox, you would get just a little dose of smallpox, and that would make you immune. And that worked. I mean, it worked largely because it was done to young, fit, healthy people who had got a good immune system. So if they got a little bit of smallpox, they could fight it off. Whereas if you were 40 or 50 and your immune system was not so good, if you gave a little bit of smallpox, it would become a large amount of smallpox and then kill you. So that is where the vaccine story started. I mean, vaccinia is smallpox. It's the same word. But the vaccines that people really wanted were actually, the first one that was really wanted was rabies, because there were a lot of mad dogs around, and rabies in cities was a significant problem. so that the Pasteur worked out that you could grow viruses. I mean, you see, you couldn't see them. The microscopes in those days weren't good enough. So you couldn't actually see the virus particles. But he worked out that you could, he could put extract, if you like, of rabies into cells growing in a lab. And getting cells growing in a lab were one of the first things that he invented, if you like, was being able to continually grow cells in a lab. So he put cells in, he put virus in, and you could make more virus, and then he killed it and put it into patients. And
So you could see cells.
yeah,
So
he could
it's
see.
like
He could
like
see
cells,
cells. Yeah, he could see cells. He couldn't see the virus, but he thought there was something there, and he infected the cells with the something, which was just the liquid off the top of cells, and that allowed the cells to make more virus. He then killed the stuff with heat and used it and put it into people, and lo and behold, it stopped the spread of what was then a very serious disease, rabies. So that was the next virus that a vaccine came along for. So it's interesting to note that these are both viruses. They're not bacteria that the vaccines are against. We're not very good at making vaccines against bacteria. Bacteria are much smarter than we are. They can fight the immune system, but the viruses can't. If the cells get infected with viruses, the body's defences against that is simply to kill the cell. If you kill the cell, there's no more viruses coming out. So that's why viruses were top of the list. And then after that, well, there was a whole string of them that some of which worked and some of which didn't. But probably the most important one was polio, and that was in the 1950s. And Salk and Sabin fought it out as to who had got the better vaccine. But as it happened, both of them worked perfectly well. And that by that time it was understood that there was something in the blood which neutralized viruses after you'd been exposed to the virus itself or a dead virus. And the proteins that we now call antibodies, were sort of fingerprinted at that time. They didn't really know how they were put together, but they knew that there was an ingredient in the blood, a protein, that could protect you against virus infection. So that the science worked backwards. You got the vaccines first of all, and then you started to work out how they were working and what was actually happening. So it wasn't a planned event where we did all the basic science, the applied science, then came up with a vaccine. It was very much the other way around. Came up with a vaccine, and that's how the heck did that work? And I think that's actually one of the more important lessons in science, is that if you can solve the problem, then you can probably work out the science behind it. We nowadays we like to think we're being analytical, and we start from let's understand how cells work, let's understand how the body works, and then we'll be able to solve these diseases. But it might be easier to turn the other way around and say, well, these people don't get cancer despite the fact that they've smoked or done whatever, so they've got something that protects them. And that has actually led to a whole lot of understanding of how the immune system works and how the immune system can help prevent cancer.
How does that research work? You go with the bloods, or?
Yeah, well, you can look at putting cancer into an animal and then seeing how the animal fights that cancer and what's happening round about the cancer cells, and you come up with this long list of cells which the body makes in response to a cancer, starting with macrophages, which were the first cell type that was really recognized with Metchnikoff's work. But subsequently we had antibody-making cells which didn't seem to do anything much for cancer, and then there were these killer T cells which were found in the 1980s. They really only became understood because we had monoclonal antibodies. This ability to make an antibody which was absolutely and exquisitely specific for just one protein, because the lymphocytes in the blood were thought to be all the same, and appeared in cancer, so people thought, well, maybe they might be involved in cancer. But we didn't know much about them until we got these monoclonal antibodies and found that lymphocytes weren't just one sort of cell. They all looked the same. They were round and small, but it turned out that there's now, well, we're somewhere up about 50 different sorts of lymphocytes, but the first two were the ones that were really important because one of them was a killer T cell and could kill cancer cells. And the antibody that identified that found a protein called CD8. And so these CD8 positive cells were lymphocytes that could kill cancer cells. They had to be taught how to do it, but the point was that they were able to do it. And
How do you teach them how to do it?
vaccines, yeah. It always comes back to the same thing. Tell the cell what to do and it'll learn the trick and then do it. We haven't really exploited that as much as we can because by the time a tumour's there, the tumour is quite able to fight back against the killer T cells. But in an animal model you can show that killer T cells work extremely well to kill cancer cells. It's all the...
Have to know that they're
Yeah, you have to know the cancer cells are there, and before the cancer gets the sort of wall of armamentarium round about it, which it puts out for, it's not entirely obvious why. It could be that it is actually part of a physiological mechanism to try and control the cancer, but we only see the ones that fail. It's frustrating because it's very, we probably each carry cancer cells around inside our bodies, and then we've got all these nice tools that nature's given us to fight these cancer cells. So we kill all the cancer cells we can, and then what you get left with is a cancer that the system doesn't work for, and you find all these cells round about it, and you think, well, they're interesting cells, maybe they're doing something for the cancer, but then you think a little further and say, well, but they're there and the cancer's there, so they're obviously not doing the job.
And who knows what you missed out? The fun you watched, the fun you failed to see while the body's been fighting off the cancers successfully.
That we get cancer cells, and one of the ways we know that can happen is simply if you get your immune system damaged in some big way, there's a very much increased risk of many cancers. So that the immune system already knows how to fight cancer. It doesn't do it 100% effectively. Partly, as I said earlier in the conversation, there's no particular reproductive advantage in stopping a cancer at the age of 60. If it can stop them at the age of 10, then you'll reproduce, and therefore the network will work, and you'll acquire the ability to kill the sorts of cancers that might go for you when you're 10. And the ones that we fail on are the ones where the immune system doesn't go, brain cancers. That the kids get brain cancers because the immune system can't get in there. The brain is well defended against that. But it's likely that we fight off cancer cells throughout our life. And as we get older, the systems get older too and don't work so well, and therefore we increase our risk of getting cancer. Of course, we do other things to increase our risk of getting cancer as well. We're not particularly good at looking after ourselves, but there is a built-in defense mechanism against cancers, which we're beginning to get to grips with and understand how it works and when it fails.
Well, actually, I want to recap again your research. Your path in research, what made you choose it, and of course your breakthrough, but I'm just trying to compress it in the way you explained it. Is that possible, do you think?
How did I end up working on cancer?
And your success, your breakthrough. I mean, I like to think you had
we were very fortunate. I mean, we grew up in an era where all the tools that we needed had just become available, the molecular and cellular tools, so that it was known that papillomaviruses could cause at least one cancer. Harold Serhausen in Germany worked that one out in the late 1940s. And so our interest was in how you might prevent or treat cancers caused by papillomavirus, so that we couldn't grow the virus in the lab. that was a bad problem. You know, if you want to make a poliovirus vaccine, well, you grow poliovirus in the lab and you kill it and it becomes your vaccine. We couldn't grow papillomavirus in the lab. We still can't. So we had to work on the basis that we could build something which looked like papillomavirus, and the body's defences against infection would say, that looks like papillomavirus, let's fight it. So that what was necessary was to actually build the virus from scratch. So we started with the capsid, the coat of the virus. We didn't really want to have to make infectious virus. If it was a cancer-causing virus, we'd probably not be very popular if we started making the whole virus. But we thought, well, if we made the coat of the virus, the body's immune defences know how to deal with the viral coat. They make antibody, and the antibody stops the virus or the viral coat from getting into cells. So that we had to start from scratch with that. We had to take the genetic information of the virus and take the bit that we thought would make the coat of the virus. I mean, all the proteins in the virus had to come from the virus's genetic information, so that there was only 6 kilobases of genetic information. It's not a very large amount. Some viruses are 100 times more than that. But we had a fair idea that that there would be bits of the genetic information in the virus that would code for the coat of the virus, and we looked for the proteins that the sequence in the genetic information of the virus that might do that, and worked out that there was this protein called L1 which looked like it might be the right candidate. And so we couldn't use the virus itself to make the protein. I mean, it was a dangerous virus, it caused cancer. So we put the genetic information into other viruses to see if we could make those viruses make the viral protein. And they could make the viral capsid protein, but they didn't assemble correctly. So they didn't get something that looked like the virus. We just got if you like, a blob of protein. And we had to eventually work out that what we really needed to do was to get just exactly the right bit of the genetic information that would produce a protein which would self-assemble to make the whole capsid of the virus. So that 360 copies of the protein assembled into making the shell of the virus. And that took about 6 months of hard work. And at the end of that, we got some bits of things that looked sort of like the virus down an electron microscope. We could see them, and we could show that those bits were immunogenic. In other words, they could be put into another animal and it would make an antibody. It would make an immune response. So that was we thought, right, that probably would work as a vaccine, and we went and sold that idea to a number of companies that could do in big scale what we could only do in small scale in the lab. Now we would be able to do it in big scale in the lab. The labs have got the technology that enables that to be done, but in those days it was just a case of do it in a test tube and see if it worked. So that once we got some evidence that it was working, it was making virus-like particles, and those particles made antibody which could recognize the virus. We then went to companies who had the capacity to scale that work up. Merck and GSK were both interested in that area. We talked about the work we'd done at scientific meetings, so we came to them and they came to us, and basically we partnered with them for quite a while, for maybe about 5 years, until they got their technology worked out. And then that's basically how the vaccine came about. They used our rather primitive approach to it and then scaled it up to make what now is a very effective vaccine.
So you're fairly nimble in what they in comparison to what happened in the past, I guess.
Yeah,
I guess they're getting faster and faster.
Well, they knew what they wanted to do, and they could see that there was a market, if you like, for a vaccine that could prevent cervical cancer. But it was all done in their research laboratories. You see, that's the interesting thing, because Merck had two divisions. There was Merck Research, and then there was Merck that makes vaccines. So that it was actually the work of the people in the research laboratories, particularly Catherine Jansen, she was determined she was going to end up with scaling up that process to make enough to see if a vaccine would work. And that's what they did. And they did it with, first of all, with animal papillomaviruses and then with the human ones, and showed that not only could they scale it up, but yes, it did protect, which was, for us, I mean, that was 10 years after we did the original work. You know, we showed the virus-like particles could be made in small scale in 1991, and it was 2001 before they got to the stage where they were able to show that in humans these virus-like particles could produce antibody, and then they went on and did larger clinical trials to show they were effective.
They showed that they were effective. It's a bit of a breakthrough, though, isn't it? Especially the viral consequence with cancer. Or
Yeah, we need a few more successes like that.
yeah, that'd be nice.
Yeah.
How's the ACRF helped in research in general? I was going to ask you, what's your lab do now? I mean, because it's a huge breakthrough, and it's refreshing to hear about a huge breakthrough, which is the reward for years of research. So that now springs into this laboratory. I wonder if you follow up there. How's the ACRF help?
Well, the ACRF has been very effective in listening to our request for high technology that we simply can't afford otherwise. I mean, they built us a lab across in the hospital, which was really important for getting the papillomavirus story more broadly known, but also it enabled us to start working on other viruses that were likely to be causing cancer. So they gave us the tools, they gave us $1 million, and basically we used that to fit out a new laboratory over there which had all the technologies that we wanted. Subsequent to that, they have partnered with universities here and elsewhere to provide particularly expensive bits of equipment which no one place could ever afford to buy. So there's a whole range of technologies which we use routinely now. We can image tumours growing in live animals to see what's going on and how the body is interacting with that. So you've got a living mouse anaesthetised, and you've got a microscope which can look at individual things within that living mouse and see what's going on, watching the cells moving and how they're killing cancer cells and this sort of stuff. It's pretty impressive technology, and they have given us access to equipment where we can make genes and access to equipment where we can take genes and modify them and then see how those genes work, so that the genetic information that we had to pull out with considerable difficulty from samples of tumours in the early 1990s, now routinely we can extract that sort of information from tissues, sequence the genetic information there, sequence the messenger RNA, so that not only what the backbone of the cell's capable of doing, but also what it's actually doing at that point in time. And you can do that for individual cells surrounding a cancer. So these tools are now available, are really very impressive. I mean, my postdocs and the people in the lab, the students, they can do things that we couldn't even have dreamed of 30 years ago. And that's possible because first of all, because people have recognized that a machine that can do that sort of thing would be really useful. And then the money from the Australian Cancer Research Foundation enables us to buy such equipment and keep up to date in what is obviously a fairly competitive race. I mean, Australia pulls well above its weight in research in a lot of areas, particularly in cancer. I mean, we're competing with the United States, we're competing with Sweden, with Germany, lots of places that are doing work. If you're going to be useful, you have to have the latest technologies, the ones that you need that will move things forward fast. And it's a sort of mixture between, well, we don't want to give everybody everything because that's going to cost a fortune, and we don't just want to have one lab doing it because that stifles competition. So that we're all involved in a big race, and we've got each scientist has a different goal in mind. But in our lab, almost everybody is looking for ways of sorting out the cancer cells before they spread through the body. You know, when they're there and growing, but they haven't yet managed to take root all over the place, then the question is, what do they have to do to move out of where the cancer starts, and what can we do to stop that? And the answers are different for every cancer. But one thing is clear is that it's very much a case that the immune system has cells in it where their primary purpose is to stop cancer cells spreading. And if we can keep those cells alive, well, and doing what they should do, then even if you have a cancer, it may well be possible to treat it, either completely get rid of it or at least contain it so it never kills you. And it's that sort of
It must
goal.
be a very exciting time. I mean, given the history of medicine, don't get me started on the history of medicine from 3,000 years ago
Yeah.
to today and the acceleration right at the end.
Yeah.
Is this not the most exciting time to be in medicine, into research,
Oh, I think it's
in human history?
we have more capacity to do things now than we have ever had in the past, and we have more questions that we need to answer because we keep making life difficult for ourselves by not looking after ourselves as well as we should. But yes, it is a great time to be a scientist because there are all these tools available that allow you to answer the questions that we could only guess what might be the answer 20 years ago, 30 years ago. I mean, it is just a case now that if I want to find out what's going on in that one cell in this particular moment, I can pluck that cell and I can actually work out exactly what that cell's capable of doing right now and whether it's doing it properly or not. And that concept just sounds so unbelievable when I look at how things were 30 years ago. I mean, that's the bit that's really changed. It's this huge ability of technology to start to answer all of the really important questions. The one bit that we're stuck with, that we haven't really managed to get there with, is the brain.
Does AI help? I suppose it must in some ways.
Yeah. Yeah, it does. I mean, we generate. Yeah.
Sorry.
Will it cut for Steve?
I think
No, we're not cutting. Steve's furniture will move away. I said, yeah, I'll take another take. You haven't changed that much from astrophysics. It's all going where no man's gone before in some aspect.
Look, I am
were you
still
a science fiction?
highly interested in astrophysics. I would really like to know the answers to some of the questions I have in that area, but the technology ain't there. You know, is it important to know? Probably not. I mean, we have not been bombarded by other alien species coming to land and make life home here, and I don't think that will ever happen. But the distances are just unbelievable. Out there must be life of some sort in many places, but I mean, because the chemistry is the same. You know, there are still just the 100 and whatever atoms that there are, and they still behave the same way as they do. We have no evidence that out there they behave differently.
Well, but to put a romantic spin on it, your universe is now internal rather than externally looking inwards, but still another universe, isn't it? And you're still being curious about it.
Yeah, look, I think it's the art of being the art of surviving in science is to be curious. If it ever becomes boring, you shouldn't be in science. You know, if you've got the wrong project, or you've got the wrong people around about you, or you've got the wrong ideas. I mean, you really have to come into work in the morning saying, "How on earth is this working? What can I do to find out the answer?"
Well, you know, is that what you love about? What do you love about your job? About research?
Yeah, well, that's it. I mean, you're challenged. You know, here's the problem. Now, what's the solution? Go out there, find it. Obviously you get me. You become excited because other people are making breakthroughs all the time round about you. Things that we wouldn't have even thought of are now routine, and we understand an awful lot more about how our body works. We also know an awful lot more about how our universe works and about how the planet works, and you know, all these things. These are questions that it can only be of benefit to us to know the answers.
Were you a science fiction fan?
Yeah.
Just so I wasn't alone.
Yeah, no,
Who's your favourite
oh, I've got to be careful about what I say about that.
And incidentally, we'll show you everything we shot, and if you don't want something in,
Yeah.
we won't put it in. So rest assured, we won't do anything like that. Just interested as a novel.
Yeah, I'm trying to remember the name of the author of the book. I mean, whose books I've got a whole shelf. James, he's a Scot, and he also wrote a great book about whiskey. No,
I like it already.
It was a whiskey galore.
yeah,
Brian Holmes.
Brian Aldiss. Yeah, no, that's not right.
Do you remember the name of the book? And we all sci-fi freaks.
Yeah. It's annoying me because I can't remember, but if you give me 2 minutes, it'll come.
Was it our generation? Because we all part of the
Yes,
space
it's all our generation. I mean, he lived in Fife. He died about 2 years ago. He lived in Fife in Scotland, and he wrote a great book about whiskey as well. But most.
It's a couple of Scots, but we are digressing, but I don't mind. If you don't mind.
I'll look it up. I can easily find it that way.
Who's the Brits kept on? Yeah, we can cut it.
Cutting.
Okay, that's fine. Just everyone seems to be when we. What's going?
Sweet.
Sorry, guys. The space race, Apollo, everything that happened then when we were growing up, everything was optimistic.
Yeah.
And it seems by and large in the last 15, 20 years, everything seems to be pessimistic except for science. There seems to be a lot of optimism coming, which I respond to. I enjoy seeing it and the people respond. Do you see the same way?
Yes, it's interesting watching the space race, and the race was run and won, and now there isn't a next step that makes logical sense. You know, I suppose that sometime in the future someone will find some way of exploiting the solar bits of the solar system that we can actually reach. But right at the moment there isn't an obvious need for, or indeed a logical purpose to getting there and doing that, which is a bit of a shame, but that's the way it works.
But that sense of optimism seems to exist within the sciences, especially within medicine and cancer research.
Yeah, I think looking inward rather than outward is sensible. I mean, we've got enough problems to solve without worrying about what's out there in space. I think that people will continue to be interested in astrophysics, as I once was, simply because there are some very definitely unanswered questions out there. But whether those answers, once we've got them, will make us feel better, that's another matter.
You can't control that.
No, that's true.
I was going to try and cover some Hippocrates and up together, which we started conversing about before, if you're up for that one.
Yeah, sure.
The history of medical research. It sucked you in the end, it got you with a lot of people, but it wasn't always a cool thing to do, was it?
Yeah, well, look, there wasn't really much medical research at all. There was an understanding of anatomy and to some extent physiology, but the driver for medical research was disease control, and the aim was always there to understand why the diseases occurred, and so viruses were the first thing that were thought of particularly, but they didn't really realize what a virus was. It was just a word, but they could realize that there were things that spread that way. And smallpox was the catalyst for understanding that. But life science is big. I mean, we made only limited progress. I mean, if you think about it, we did blood transfusions not knowing what blood transfusions were all about. I mean, we found out pretty quickly that if you gave the wrong sort of blood.
Transfusions sort of
Well,
we're talking 1800s.
we're talking early 1900s, when they found out about blood groupings, because that was the first thing that they needed to know. They worked out pretty quickly that if you give blood to somebody that wasn't the right blood, then they died. So that they worked out that there was a problem with compatibility. That was in the 1910, 1920 period. I mean, blood became important when
Beeping machine.
the beeping machine, yes, just to annoy you. Knowing blood typing became important when surgery became possible because of anesthesia. I mean, there wasn't much need for. I mean, in the old days, if you had a bad leg, they chopped it off pretty quick, and there was no attempt at anesthesia until they got ether, and then you could start to do planned surgery, and then you could need to know about blood grouping because you were going to spill a lot of blood, and you couldn't just take it from anybody, as they found out pretty quickly. So usually the progress in science follows mistakes. You do something wrong and you learn from your mistake. So I think that's true particularly in medicine. We're still pretty primitive in our approach to medicine. You know, largely because we're trying to fix problems that are already there, where we should be trying to prevent them before they happen. You know, we go out in the sun and get skin cancers. We know that going out in the sun will increase your risk of skin cancers. We still go out in the sun and get skin cancers. We eat a diet which has got too much fat and too much sugar in it, and we get heart disease, and we know that that will give you heart disease, and we still go out and eat too much fat and too much sugar. We let people become overweight. We don't. There is a push to give people sensible, healthy diets, but the sensible, healthy diets turn out to be more expensive than the cheap, high-calorie foods that we can easily make.
Possibly less tasty.
Yeah.
Plus there's no salt and sugar.
Yeah, well, we like the taste because we're genetically programmed to like the taste, because if you're going to live in a place where there isn't much food around, then having an enthusiasm for the high-calorie content foods is a good idea. You know?
Is there anything you want to talk about? It's your journey as well.
Well, I think that
I mean, I know you've been interviewed to death, but there's things you've always wanted to say that you've never said.
Look, I think that we don't encourage people to take a career in science like we should. There are so many problems that we could deal with if we put the effort in, and in health particularly, but not just in health, and cleaning up the environment and working out how best to deal with the fact that we live on a planet which doesn't always give us the weather we would want. All these things, we could put a lot of effort into that, more than we're doing. Instead, we put it into, well, what are convenience things? I mean, cars that go ever faster for no particular purpose and carries 4 people around, when mostly people just go take one person or should take the bus. You know, we could live in a much more, how can I say, efficient isn't the right word, a society that was much more likely to give us all a good time. You know, we starve 1/3 of the world's population. There's plenty of food around. It's just not in the right place. We are not good at working in teams. look at how government works or doesn't. We need to understand the social sciences and what what is the impact of overcrowding? What is the impact of a significant number of people who are not well integrated into society? We could spend a lot of time and effort and probably end up with a better place to live. I mean, the built environment, looking outside the window there, there are some problems. There are going to be even bigger problems once they make their mind up what they're going to do about the Olympics, but that's...
Oh, that's local problems.
Yeah, but I mean, that's what I'm saying. The built environment is a local problem. There are 4 million people live in this corner of Queensland, and I don't think the built environment is in any way favourable to them. We try to preserve bits of it to make it. I mean, we know that an environment where there are open spaces where people can move around freely, where it's not a concrete jungle, people are healthier. They generally will be happier, and there'll be less violence. It's not accidental that most of the crime occurs in the inner cities. We need to make a life for people that makes them comfortable. We also have to decide what size of population we really want to have. I mean, we're 8 billion now.
Has it reached 8? I thought we were still in 7, but I'll trust your numbers more than mine.
Yeah, and that seems to be too many. I don't know if that's the right way of looking at it, but I think we could probably have a more comfortable life on this planet in the future if we decided that the population was to be 2 billion rather than 8.
Well, I agree with that, but I was saying that you said this in 68 to 70. I was living in Brisbane and I can't recognise that city to what I'm seeing today. It's an incredible change.
Yeah, well, it's changing rapidly, and I mean, I came to Brisbane, and we had a rush half hour in the morning, or maybe a rush 10 minutes, and now we've got traffic gridlock for half the day. And do we really need to go by car? We've got a great bus service in Queensland. The buses are actually used quite a lot now. We only have to pay 50 cents a go.
Are the trams still running?
They've reintroduced trams.
I've gone off piste, I'm sorry. Steve, do you have any questions you want to ask him?
Yeah, I'm very interested.
Look at me when you answer.
Yeah.
What he was saying that, this one point it's discovered that viruses had some relationship to cancer. And I just was wondering, who was it or how was it that we take viruses and cancer? Because we found to have some connection, because I believe that a lot of virus is interdependent with cervical cancer.
Yeah. Look, funnily enough, the first virus that was shown to cause cancer was the papillomavirus, and it was canine oral papillomavirus, a dog virus. Dogs bite each other, and the virus spread, and they could see the virus on the electron microscope, and they worked out that was probably causing cancer. We don't have a vast list of virus-induced cancers. Most cancers are not virus-linked, or if they are virus-linked, we haven't found the right virus.
What percentage would you say?
Oh, look, I mean, if you take cervical cancer out of the equation, it would be less than 1%. With cervical cancer, it would go up to about 3 to 4%. I mean, the cervical cancer is the 9th commonest cancer, and it's entirely caused by papillomavirus. But all the ones further up the list, there are environmental factors that are the major drivers. Lung cancer, obviously. And then there are the genetic mistakes cancers, where you inherit the wrong set of genes by misfortune, and then end up with a cancer consequent to that. So that it's nice to know that viruses cause cancer. It's equally nice to know that most cancers are not caused by viruses, because then that drives research to see what else we could do to reduce the burden of cancer. But we know what we need to do. We need to get rid of environmental toxins. We need to eat a sensible diet. And if we did most of the simple things like that, then we'd gain about 2 to 3 years of life on average. You know, it's not, that doesn't sound like a lot, but it's it is quite a lot.
Fair enough. I've taken up an hour and a half of your time. So.
Well, I hope it's been useful. I think that.
Yeah,
Sweetie,
Yeah.
slide it.
then slide.
Yeah, so the viruses that cause cervical cancer cause a precancer before there's a cancer. And that precancer, these viruses are interacting with the cells, and in many people the cells fight back and can stop the virus causing problems. But why only 1 in every 50 people who get these viruses go on to get a cancer is a really interesting question. Is it because the virus is a different one for them? Doesn't seem to be. So if it's the same virus, we must have natural defenses against the virus that causes cervical cancer, and that some people have them and other people don't. And that's still an open question. We don't know. We know that quite a lot of people get the virus, have the infection for a short period of time, clear it, and no problems. That's almost certainly simply an immune thing, you know. But then there's a group of people who get the virus. It causes a precancer which lasts and may last your entire lifetime. Doesn't go on to cancer, doesn't regress. So there's a battle going on between the host and the virus. And if the virus wins, then you get a cancer. And if the host wins, then you get rid of the virus. But in between, there's a battleground. And it would be quite nice to know exactly what was in that battleground and what we could do about it. Because it might give us some clues as to how we could create the same protection for other cancers where there wasn't a virus involved. Because we know there are pre-cancer conditions for many cancers that long before the cancer spreads, there are cells which are on their way to being cancer. And we don't get rid of them, and if we're lucky they don't progress to the cancer, but if we're unlucky they do. So understanding that interaction would, I think, be very helpful in reducing the burden of cancer.
Well, yeah, reminds me, there was a documentary I saw in the early 90s. A guy who was like continually exposed to HIV and just didn't know it.
Yeah, I looked after one of those patients who had HIV. He had lots of it in his blood, but he never got any disease. So he was lucky. I mean, he had the right set of defences.
Which we still haven't identified.
Yeah, we haven't identified. It's not antibody. It's not that he's making an immune response against the virus. It's something else. His cells have learned a trick, if you like, that stops the virus doing what it could do.
Something else out there.
Yeah, well, it doesn't stop the virus replicating. So these people who carry the virus continually can pass it on to other people, and they get the disease. So it's not that the virus is different, it's that the host has got the measure of the virus and keeps it under control without actually getting rid of it.
That'd be a nice thing to know.
Yeah, well, knowing, look, everything we learn is nice to know. You know,
I've put a bit of myself. Yeah, that was, thank you. 5 favourite discoveries in science.
Well, I think that the most important discovery is that if you don't provide the money, the research doesn't get done. And if you can provide the money, then people will be motivated to go solve the problems.
That's 1 through 5, actually.
Yeah, precisely.
Okay. And Slade?
Sounds like you can go get some house, please.
computers really. I'm elevated to that. 4?